Hi, Jason. Hi, Claire. Can you hear me?
Hello there.
Hello, hello. Can you see me? Can you hear me okay?
Yes, I can see you and I can definitely hear you. How are you?
Fantastic. I'm good. I'm good. Thanks. How are you?
I am very good. How has microbiology day been coming along?
It's been going well. Yeah. It's we've had a lot of diverse and interesting topics being covered here, so it's been really fun. I'm definitely looking forward to this session now.
Fantastic. If you're good to go and everybody's happy, we can just get started.
Yeah, we are good to go. I'll basically hand over the floor to you, Jason.
Thank you.
Yeah, it's yeah, and I'm here if you need anything in the background. But yeah, Jason Tetro. I mean, best-selling author, host of the super awesome science show. Take it away. We're very happy to have you.
Well, thank you very much. And thank you everybody for being here on this amazing International Microorganism Day. And I have to tell you something, when Ben asked me to be a part of this, I was just super thrilled, as I normally am when people ask me to do things live. Normally I do stuff taped, my podcast is taped. But there's something about doing something live that is just amazing. It feels like you're always doing an experiment. And for those of you who are in the lab, you know that feeling, right? You got the nerves going. You're kind of like, oh, I'm hoping it's going to go well. It's really awesome. But I do want to tell you something. And I mean, I know it's a late night talk show and it's we're supposed to be happy go lucky all the time, but I'm kind of feeling a little bummed out. And you know what? Why don't I just show you why I'm feeling a little bummed out? Okay. You see, we want to be living and loving our microbes. And Ben, can you see that? Excellent. And so, one of the things that I've been doing for so many years is just trying to get people to sort of figure out how to have that relationship. Now, you have to understand something. Relationships are hard, and I totally understand that. We have so many relationship therapy models: getting the love you want, he's just not that into you, therapeutic attachment. The reality is, the goal is really to best describe how the relationship exists and then figure out how you're going to be able to do that. And when I started writing my books, that was the first thing I wanted to do. And yes, the books are really a how-to on how to love germs. You got to get used to it. And really what it came down to was, if you remember in 1991, there was this wild book. It was called Men Are From Mars, Women Are From Venus, right? And what I did is I thought about it and I thought, okay, well, if we are humans and we're from Earth, well, where would germs be coming from? And we thought Mercury, maybe Jupiter, Saturn, Uranus, or maybe the planet formerly known as Pluto. Still upset about that. And we realized something. All you really need to do is just change the way that you pronounce something. So humans are from Earth and germs are from Uranus. And for those of you who don't speak English, it really is funny. Just believe me, okay? Anyway.
It is. It is.
But the fact of the matter is that despite that microbes are in our GI for the most part, they do happen to be everywhere. And we do have this microbiome. And I think the one really cool thing about the microbiome that everybody really needs to understand is that the more that we get a feel for what the microbiome does for us, then we can understand how we can work with the microbiome. And a few years ago, I actually co-edited a book on this, the Human Microbiome Handbook. And at the same time, I was actually writing the germ file. So you've got the academic version in the one book, and you've got the public version in the other. And the whole idea was that whether you're a scientist or whether you're just someone walking off the street, you really want to figure out how you can create a love story with germs. And let me tell you something, it was going well. It was going so well. And then this guy came along. I'm sure all of you know who that happens to be. SARS-CoV-2, also known as the causative agent for COVID-19, coronavirus disease 19. Well, this thing just kind of messed everything up. It's a late night talk show, still not going to swear. But what ended up happening was that the masses of people who truly were starting to understand the relationship, truly understanding how we can love microbes, started running away. And where did they run away to? Well, yes, the underground, otherwise known as we hate microbes. But I'm showing the underground for a different reason. And that was after COVID came out, there was this very interesting paper that was published in the New England Journal of Medicine, which talked about how the virus could possibly survive on surfaces for a very long time. Interesting, I used to do the exact same type of research. It was a laboratory controlled study, but it kind of blew up in the media. And all of a sudden, well, we had people wandering around looking like Ghostbusters, except they were germ busters. And they were going around spraying everything. And the whole point was to try and spray everything that we were touching in the hopes of getting rid of COVID-19. There was only one slight problem with that. I know that we're probably blocking it a little bit, but if you look over here, you're going to see a little animal holding a hand sanitizer bottle. The reason this is important, that's a Banksy. Now remember, Banksy has sold paintings for like millions and millions of dollars prior to shredding them. But anyways, and the fact is that they actually got rid of Banksy paintings while they were doing this hygiene theater of trying to disinfect surfaces. It was a nightmare. But more importantly, it took away the one thing that we should have been paying attention to, which was the fact that the rats were showing us what we really should have been afraid of. Now, when I first showed this, I actually called them droplets. We now understand that it's probably airborne. So I'm just going to use the term aerosols, but by the same respect, it all means the same thing. Surfaces, airborne, absolutely. And of course, we did eventually figure out how to protect ourselves, right? And of course, you know, it is England, so they're going to protect themselves any way they possibly can. But the fact is that you have to understand that when you see this happening, you realize, okay, I've got to start thinking about how I'm going to be able to protect myself, but also do it in a proper way, which is, of course, the whole idea that I'm talking about when we're falling in love with our microbes. And here's where it gets really, really scary. I don't know about you. Have you ever taken out something from a restaurant and it goes into this takeout bag, otherwise known as a takeaway bag? Did you know that this poses a risk for you to get COVID-19? Why? Yes, according to that New England Journal of Medicine article, it does. But let me tell you something. If we do a risk assessment, it's a very low touch surface. It has no contact really other than the handles. And it doesn't really know, it's not really known to have any surface transmission from one person to another. But the most important thing you need to know is that it's not the bag itself that poses a problem, it is the food. Foodborne infections are the most common type of infections that we face. And that's one of the reasons why we have food hygiene ratings when it comes to restaurants. But I don't know about you, I've never seen a takeaway bag rating. This is what it means to have some kind of relationship with microbes. And just so you know, if we do end up actually getting a takeaway bag rating, it probably will be Germ Guy approved. And that's one of the reasons why I find myself so bummed out. And now the really interesting thing that you have to think about is the fact that when we do talk about our relationship with microbes, we do figure that it really is about us and the species that cohabitate that three-dimensional space with us. And we are going to get into that a bit later. But I do want to ask you all whether or not you believe that these microbes can have an effect not on our human to microbe relationship, but our human to human relationships. Now, this is a question that I asked someone a while ago, and it was in a very different context. But what I did is we talked about it on the show, and it was one of the best segments that we ever had. Like, it was just so good. I had people writing me going, oh my God, who is this person? We love her. And thankfully, not only was she loved in Florida, but she was also loved in the great state of Tennessee. And so I've asked her back for this particular show so that we can have a discussion about the aspect of relationships that germs have, not necessarily with us, but what the effect is on us and how that creates a relationship with others. And it's for one very important thing. Are you sure? It's the way we smell. Please help me welcome to the program, Middle Tennessee State University Assistant Professor, Dr. Jessica Gaby. Hi, Jessica.
Hi.
How are you?
Great. How are you?
I am absolutely fantastic. Thank you so much for joining us on the show. I mean, the last time we talked, we had such a great time. And honestly, you were the first person I thought of when it came to having this show. And so I'm so thankful that you were here and you're able to join us. I know it's a little bit late, so also thank you for that. Now, one thing I do want to bring up is that normally when I introduce people, sometimes I give them a little bit of a background on the history, et cetera, et cetera. And I just called you an assistant professor. And the reason I did that is because I want to start off our conversation with a very important point. You're not a microbiologist.
I am not.
Tell us, no, exactly. So tell us about your research program.
Yeah, so I study how the way that we smell impacts our social relationships with one another. And specifically, I'm interested in platonic relationships. So a lot of the studies that look at body odor have been done on mate choice, but that is such a small percentage of our daily interactions. I'm really trying to look at how does the sort of odor personality that we build for ourselves in real life impact other people's impressions of us. And so I've looked at that from a couple of different perspectives. I've looked at how perfume might impact it. I'm currently working on a study looking at how diet might impact that. And
I mean,
yeah,
come on, diet should already do that. Regardless of microbes, you eat garlic, it's going to be hard to really,
well, see,
make
you
out
would
with
think
someone.
that. You would think that, but the only study that has been done on the influence of raw garlic on body odor showed that people like the body odor more after people ate raw. It was men. It was women smelling men. So again, caveat that it's like a specific subset of judgments, right? But they found that the body odor of the men was rated as like more pleasant and more attractive after they ate, like, I don't know, I think it was something like 4 cloves of raw garlic.
I almost feel like I should have read the book Perfume before I started talking with you again. But before we get into any of the discussions about that, I do want to go back a little bit back to your PhD thesis from 2017. And the reason I want to do this, people, is because this is what it actually is called. Invisible Influence, the Role of Human Social Olfactory Cues in Ecologically Relevant Interactions. I don't even know what the heck that means, but I love it. So can you please just sort of take us through what that means and what it was that you actually sort of found out during that research?
Yeah, so, I mentioned earlier that I study these platonic interactions, right? And one of the main reasons that I do that is because I'm really interested in how these cues are functioning in real interactions, right? Because the majority of body odor research has focused on axillary sweat, so armpit sweat collected on either t-shirts or pads. And then sometimes it's even the body odor, the sweat of like multiple individuals combined. And when you come into the lab, so when you come into the lab, what you're smelling is just armpit sweat. And you would have to know someone pretty well to get unadulterated armpit sweat, because this is not just regular armpit sweat. I asked you to stop wearing deodorant for 2 weeks, and then I took your armpit sweat, right?
This is giving me memories of that movie Flirting with Disaster, where the guy literally had a fetish of licking armpits. So
well, you mentioned that movie Perfume. If you want to get into like weird small fetishes,
that's a good point. Yeah. Also, late night television, guaranteed. Okay.
But yeah, so my sort of interest in body odor revolves around, well, okay, we know the way that this body odor functions in a lab when people have investigated this very removed type of odor sample, right? If I ask you to stop wearing deodorant for 2 weeks, I ask you to change your diet so you stop eating aromatic foods, chili, curry, garlic, asparagus. I ask you to wash only in unscented detergent and soap and shampoo and everything. I provide you all the things, right? And so I've eliminated all of these odors that actually especially in America, are really important to the way, I mean, not especially in America, across the world, right?
Mhm.
Fragrances are important. And it's weird because America, the American goal for body odor is to smell like nothing.
Yeah.
But that's not true in other places. And so what I want to know is, okay, we know these phenomena happen in the lab, but does it break through into real interactions, right? And this is like a
Mhm.
whole different beast now that we're in sort of social distancing, masking territory.
Oh
So
yeah, absolutely. Now, the thing that I get a kick out of from your PhD thesis is that you were one of the first that I know of who was doing the T-shirt experiments that we're all familiar with, right? Like the MHC 1 and 2. Oh my God, are you attracted to someone because of the T-shirt? You actually did that, but more importantly, you did that with live donors. Like this
yeah,
wasn't
so
you just collecting their sweat and then putting into a T-shirt. You actually got them to sweat into a T-shirt. How does that work? Like, do you just get them to sweat and then they take them off? Do you
well,
have like sexy music and slow motion photography? Like, how does that work?
I did do a study where I had to have a lot of women undress in front of me. It's not published yet. We're trying to get more funding to extend this project so I can get more women to undress in front of me. In that study, so that study is actually a really great one for the topic tonight, but I want to just briefly cover. So the 2017 study, what's unique about it isn't the fact that we use T-shirts. What's unique about it is that we had people smell each other in real life, right? So we had a bunch of individuals in separate rooms, and they had blindfolds and earplugs, and they got a series of visitors who would come and sit down next to them. So they would cross in front of each person, sit down next to them for a minute, and then cross in front of them again and leave. And then the person would take off their blindfolds and answer a bunch of questions about the visitor that was just there.
Mhm.
And so what we did in that study is we compared, we did a couple of things. We compared, first of all, if you meet the person a second time, do you get a similar impression of them? The answer is yes. Our impressions are very consistent in this context of this, what we would term a live interaction, right? So a more ecologically relevant interaction, something that's more akin to the kind of interactions we would have in real life, where maybe you would be sitting next to someone on the bus or in class, right? And then the other thing that we did is, we compared that perception to smelling someone's T-shirt. And we asked, well, how similar is it if you smell their T-shirt versus if they sit next to you? And the answer is similar, but not completely the same, right? Obviously, there's a lot more richness of information in a real life interaction. And then the last thing we looked at is whether or not we perceive people differently if we do have them do this removal of all the external odors,
Right.
versus letting them show up the way that they really smell. And the answer is there's a drastic difference in the way that people perceive an individual if they come smelling the way that they normally smell in real life versus if they've done this, what we call a washout.
So in this, what I called on the show speed smelling instead of speed dating, do you feel that may actually have a much more influential perspective when it comes to not necessarily finding a mate, but as you said, just platonically hanging around one another?
Oh yeah, absolutely. So I got into this work because there was a person in my friend group who to me smelled like very aversive. I was never able to develop a close emotional relationship with this person because they smelled so bad to me that I couldn't get physically close to them, right?
Right.
And so I started thinking about just how important these cues are in our social interactions. And obviously there's a big range of impact for different people. There's a lot of individual differences. I'm a person who thinks a lot about smell, so it's probably more important to me than to some other people. But certainly the sense of smell impacts us on a daily basis in ways we really aren't necessarily consciously aware of. And I'll tell you the other thing, which is that like whenever I tell somebody that I study body odor, the first thing they say is, oh man, there's this guy in my office.
I mean, there's always a guy in the office, right? That sort of brings me to the next question. I want to get to the women undressing in a second, but before we do, you are so passionate about this. Like even now, just talking with you, I'm just listening, completely enwrapped. What is the motivation behind this other than to try and find out why you couldn't have a relationship with someone because of the way they smell? Like,
I mean, I think what is exciting to me about the sense of smell is that we still know so little about it. So I was sort of going back and just reading some current papers in preparation for this. And it's so interesting to see how everyone's like, well, we don't really know the mechanism for how this works exactly. Like, we don't know what it is in body odor that's important, right? We're not sure from a chemical standpoint exactly which signals they are, and we don't really know how to measure them. It's just sort of looking into this abyss of unresearched space, which is very different from most other sensory systems, right?
Mhm.
Vision is very well explored. Audition is pretty well characterized. And so the sense of smell is just wide open. And because I'm interested in this tiny little avenue, right? These platonic interactions that
Yeah.
most people aren't looking at. For me, I can even look at a study that was done, recently where they had men, women smelling men, and I can say, well,
Mhm.
okay, but does that work between women? Does it work between men? No one knows what works between men because almost
Mhm.
no one studies male-male interactions, including me. Although, hopefully it's coming with more funding.
And do you actually because I want you on the show because obviously a lot of the smells are made by the microbes themselves.
Mhm.
And that is really interesting. But I haven't seen yet anybody try and translate the microbiome to the sense themselves. And I think you kind of already answered that. We don't even know what the mechanism is.
Well,
So
so I love that you asked this because last year, well, pre-COVID, I
Mhm.
did a really cool study. I started a really cool study when I was at Penn State as a postdoc. And we looked at the impact of a dietary manipulation. So we asked a bunch of Caucasian women who reported not eating curry on a regular, like less than once a month. They had to say it
Mhm.
once a month or less. We had them eat curry for 28 days, and we took their body odor before and after they ate this curry. They had to come into the lab every single day, and they would eat just a quarter cup of rice with it's 10 grams of curry powder in it. So it's a pretty large
Did you
amount.
give
It's
them an extra
essentially,
did you give them an extra toilet roll when they walked out?
no one really reported gastric upset. So what we were trying to do is sort of mimic, okay, if you ate curry every day, how
Yeah.
much curry would you get in a full meal? And we wanted to not impact people's dietary intake because that might have its own influences. So we sort of tried to condense that into the smallest serving size possible that was still kind of, again, ecologically relevant. So we fried the curry powder in butter to make a curry paste, and we mixed it with
Mhm.
rice, which is mimicking at least some of the preparation steps that you would do if you were going to really make a dish of curry. But we really wanted to minimize the other influences, right? So we didn't add salt.
Right.
We didn't add something like coconut milk. We told people they could add stuff on their own, but we really tried to so they ate
Yeah.
curry. So these women who don't normally eat curry, we had to make our own spice bun because there's no curry on the market that's not spicy.
Yeah.
And I was worried that if these women weren't eating curry, they would also not like spicy food. I didn't think I would be able to get them to eat a
Right.
bunch of spicy curry every day for a month. So we collected their body odor on T-shirts, but also because we know or because we expected that diet would be linked with changes in body odor, we collected their skin microbiomes to look at whether or not we would see a shift in microbiome
Oh, wow.
in conjunction with this dietary manipulation.
Okay. Well, don't just leave us hanging. Did you find anything? We got 2 minutes. We got 2 minutes left. I wanted to get to the naked ladies and also to the vanilla.
So these are the naked ladies. So every time, we actually took their body odor every week, and
Okay.
every week what they would do is they would come into the lab, and I'd apply. So we were also trying to collect skin volatiles, which it turns out is just really difficult to do, and we weren't very successful in measuring it. But we would put a little skin volatile collecting strip on them, and then we would have them, they would put on a new T-shirt, they come back the next day and they take the T-shirt off, and I would swab their skin and I would take their little volatile strip. And so every week all of these women would have to come in and undress in front of me, and I was doing it in a space and anyway, what we found is that honestly our results are inconclusive, and that's why we're applying for more funding. We found a borderline influence where people who hadn't eaten the curry. Right. So we asked the curry eaters to rate each other as sort of a measure of in-group familiarity. And then we asked a bunch of people who are unrelated to the study. And we found that for those people that were unrelated, there's a borderline effect where they find the body odor slightly more pleasant after the curry. And then what we saw is some shifts in consistent shifts in microbiome from pre to post, but it's just, we need more data. We just need more data.
Honestly, being in microbiome research for as long as I have, we always need more data. we could do this forever. We're going to have to hold off for the vanilla discussion until maybe I'll have you back on the show again in season 3. But Jessica, thank you so much for joining us. I'm so thrilled that you agreed to do this, and keep doing it. Congratulations again, assistant professor. I'm just so happy for you. I can't wait to see what you're going to do in the future, because I want to find out what those women are doing with their microbiomes. But anyway.
I literally just submitted a grant for more funding yesterday, so we'll see.
R01, obviously.
I need more data first. I need more data before I can even do that. So yeah.
preliminary data. All right. Yeah. Thank you so much, everybody. Dr. Jessica Gaby.
Thank you, Jessica. That was great.
Oh my goodness. Well, as you can see, Ben, when I bring on people onto the show, they are absolutely amazing. They are just so fantastic. And again, you also see just how amazing their research happens to be. And remember, Jessica, Dr. Gaby is not an actual microbiologist. She studies from a psychological perspective, and yet it's just so amazing what she's actually able to do. Now, the other thing that I wanted to be sure of is that when we start talking about the relationships, one of the things that people have been discussing from day one is, what does make a normal microbiome? What does make a healthy microbiome? And the fact is yes, indeed, we need more research. We need more data. However, I don't know if you look at the Nature papers and stuff like that on a regular basis. I actually found some incredibly fascinating research about the persistence of microbes in the first year of life. Now, for anyone who's done microbiome research, you actually know that this is like one of the most controversial things. So if anybody actually comes up with some data that shows us what it's like in the first year of life, it's amazing, okay? Because honestly, when you think about it, we normally associate that to perhaps the mother, maybe to the family, possibly to a pet. But that's not really the most scientific type of analysis that we can have. And so when I saw this paper, I knew I had to bring her on as my next guest. And because she has been studying the infant microbiome, and her results are amazing. Now, she's going to be showing us some slides that are probably going to be able to give us a little bit more understanding. I've looked at them. They're incredibly awesome. But before we do, I want you to give a warm welcome to the University of California, Berkeley doctoral candidate, and I'm sure is going to be a PhD in no time. Claire Liu. Hi, Claire.
Hey. Thank you so much for having me.
How are you?
I'm good.
Thank you so much, first off, for agreeing to come onto the show. And I mean, when I saw the paper, it was immediate. I just needed to be having you on the show because I wanted to hear more about this. And I was just so fascinated that, not only was it years and years of work, but it actually answered questions. Seriously, if I had you around when the microbiome handbook was being written, it would have been so much better. So first off, thank you so much that you've taken this type of research. To start off our discussion, before we get into the slides, when I asked you sort of how to, what to call you and that type of thing, you actually said that you use computational biology to study the infant gut microbiome. And so before we get into that, how did you go from computational biology over here, which is really systems biology, as we used to call it when I was young and had hair, to infant microbiomes? It's absolutely fascinating. Can you sort of give us a little bit of that journey?
Yeah, totally. So I actually, this all happened when I was undergrad at Berkeley. So I stayed at my institute for a PhD. And then I have always been fascinated with healthy diet. And the one thing I kept asking myself was like, why do I have to eat fiber, even though knowing the fact that, my body can't digest that. And then I also... And then I interned at a couple food companies, and I asked people around and no one really know. And then I also struggled with stomach problem for the longest time. And it's kind of like a very kind of funny story where like I went to Tibet with my parents, and then we went to this temple and I saw this monk, and then he gave my parents this kind of kefir grain. And that was the first time I learned about kefir or kefir. And my parents actually made our own kefir at home. And I would say after maybe about 5-6 months, my kind of struggle with stomach would just kind of disappeared. And later I learned it's because like, kefir is like microbes and probiotic.
Yeah.
So that got me really excited about this whole microbiome field. And I read some books and watched this documentary Cooked by Michael Pollan, based on his book. And then I decided that I want to study microbiome. So I switched my major to microbiology and then applied for a PhD to just study infant microbiome.
It's just unbelievable. Now, as I said, we never had that kind of research when I was editing the book. So you have to understand, I am incredibly excited to find out about your experimental and analytical approach. So if you don't mind, can you bring us in and let us know what you were doing?
Yes. So I think maybe easier if I just like pull out a slide here. Let me just get my setup going and then I'll share my screen.
Sure.
Okay. So I'm all right, can you see my screen?
Yeah.
Awesome. So I'm very curious about like, what constitutes early life microbiome and where the microbes coming from, and if they come, what's their fate? And also, is there any differences between, full-term babies and preterm babies? Because we know that a lot of preterm babies, when they were born, they received routine antibiotics, so killed the germs that were colonizing, like literally the day zero. So to answer that kind of questions, we actually enrolled about 180 babies. And then in the end, we were able to follow 42 babies from birth until age 1. And then we literally collected their poop every month for the first 4 months. And then after that, every 4 months, we collect poop from them. And then we do what is called genome resolved metagenomics. Basically, you sequence the poop and then you look at who's there and what kind of things they're doing. So I was able to track what's there for the 1 year and also compare that between full-term and preterm babies.
And when you were doing this, and I was looking through the paper, you were talking about this idea of de novo constructed microbial genomes. I find that absolutely fascinating. But what I'm very curious about is, was the amount of microbial species so small that you could just simply do this, or was there an actual computational trick that you used to be able to do this? I'm really curious.
So I'm gonna show this diagram. It's a little bit complicated. I would just like briefly walk you through like the main storyline. So basically, like I said, we take the poop, and then the poop is like basically full of the microbes that live inside of our gut. And then we just use the sequencing technology. I feel really blessed to be able to do this kind of work at this era because the advancement of sequencing technology. And then we just basically sequence a bunch of DNA. And then our lab is very specialized in genome resolved metagenomics. It's basically recover individual microbial organisms straight from the poop, straight from the DNA that we sequence.
Okay.
So that's basically like everything all the way here.
That's amazing. So whenever we've been talking about the idea of, infant gut microbiome, the first species that always comes up is bifidobacteria. And I'm just kind of like wondering, is that really the case? Or is it just one of many species that are around within that first year? Like, what really were the most persistent strains that you found?
So for the persistent strain, bifido is definitely one of the group of bacteria that we saw persisting in babies. And then another group is actually Bacteroides, which is another common gut commensal. And I think it kind of makes sense to me in the sense where both of them break down polysaccharides, carbohydrates, and then fed that to downstream microbes. So they're really important. So being able to persist in the babies are also really important.
That's amazing. Now, I have to ask you a question. Have you ever actually watched the show CSI?
No.
No. It's one of these shows that goes and uses specialized laboratory technologies to try and come up with an answer and identify a killer. And one of the times that they did the show, they were actually looking at the idea of being able to identify twins because of their DNA. And I mean, that's pretty obvious how you do that. It's pretty simple. I mean, it's been around for decades and decades. But here's something that I didn't realize, and that is you might actually be able to predict twins based on their microbiome. And when I read your paper, I was like, oh, I gotta ask you about that. So please tell us the story about infants 7 and 133.
Yes. So I will quickly bring this over. So this is about this figure in my paper, and I will just focus on this particular one. So I was doing strain results study, basically like looking for the number of shared strains between all the infants in my study. And, you know, from this diagram, so the x-axis are all pairs of infants, and I have removed all the siblings. And, you know, most of the babies here share like 1 to 2 bacterial strains. And then I saw this pair here, infant 7 and 133, shared 11 bacterial strains. And to me, I was like, that is just biologically unexpected to see that amount of like, you know, high strain sharing. And then,
Right.
so we have collaborator in Pittsburgh University where they enrolled all the baby and collected the fecal sample. And they also collected all the metadata. And then we talked to them, and then they went back to like all the files they have. And in the end, they actually found that they were like, oh, this is actually a pair of siblings, and they were born 2 years apart. They just forgot to document that on the metadata. And when I found out about that, and I was like, this is just so cool, because like we also collected poops from the same mom, actually 2 years apart. So I was like, we can like actually see like adult microbiome persistent or like sibling transmission, all sorts of kind of questions, even though it's just one case.
But what you're telling me is that they're not even twins. They're
No,
siblings.
siblings, actually.
Wow. It really does make you think about it, really, that the microbial species that happen to be within an environment are really that predictable that you can pick people out who happen to be related to one another. I think that's incredible. But more importantly than that, I think it brings up this whole question of, you know, genetics versus environment. And it definitely seems to suggest that perhaps it is the environment, because they aren't twins, so they don't have the exact same genetic makeup. Like, does that make sense to you?
Yeah, definitely. I think environment definitely plays a very important role in terms of like the microbiome, who's going to colonize the gut, and also how long that kind of species are able to colonize the baby's gut. Like one thing I spend a lot of time on is maternal transmission, and how strains are vertically transmitted from the mother. How long can they stay in baby, and is this kind of transmission, you know, continuous throughout the first year of baby's life? And this is a lot of stuff going on, but just focus on like the kind of line. At this time point, it's like the first time point where we detect a particular strain. And, you know, if they are detected all the way until age 1, you're going to see them cross all the way down. Right. And these are all the strains that's maternally transmitted. And one take-home message here is just like, you can see this kind of maternal strains appeared in baby throughout different time points. And some of them, you know, they can stay with the baby for the first year of their life. And the orange one are actually referred to the Bacteroidetes, the Bacteroides strain that we found to be one of the most common bacterial species to persist in baby.
Wow. That is just amazing. But it's also so small. It's funny, not small in terms of size, but small in terms of the number. We always talk about diversity, diversity, diversity. That's the one thing. But perhaps maybe it isn't necessarily as diverse as it needs to be, but you have to have the right diversity in the gut, as opposed to just
Exactly.
a massive amount of diversity. So how many species are we talking about? Like in total, did you find really were persisting?
Oh, in total, we're persisting. So in total, I found like about 11% of everything that I looked at ended up persisting in babies. Very small amount. Baby in
Wow.
general have a huge turnover in terms of who's coming in, who's leaving, just because baby's gut is so, immature, kind of chaotic, not like adults microbiome where it's
Oh my
relatively
goodness.
stable.
Wow. All right, turn off your slides. I need to see you for a
Okay.
second, because
Okay.
I need to be able to see you in order to tell you this. We've gone through a lot of information just in these few minutes. And even right now, my head is just like, oh my goodness. Most of the time when people do research, it's really about the paper. And they just do the science to get from concept to publication. They don't really sense what's going to happen afterwards. Having been seeing microbiome research for the last 10 years for me, what you're showing is going to change perspectives. It literally is going to change the way we do research into the future. And I'm wondering for you, as a PhD candidate, who obviously is going to get her PhD after this paper, what does it mean for you to know that even at your young age, you're starting to make a difference that's really going to translate into, a different way of thinking down the road, especially for mother and infant health?
Mhm. I mean, this paper is like very, very special just because like I think it's my first author paper, and also the kind of, the kind of influence I hope to have, like to make people realize that, the early life colonization is important, because if you accidentally have the wrong kind of microbes, they might stay with you for a really long time and might influence and might have like impact on the host health. So I would say during the writing process, I definitely went through a lot of challenges. Like half of the data is contaminated. So I have to throw half of my data away.
Yes.
And like you said, we need more data. And I totally felt that like losing half of the data really changed a lot statistically. And whenever I felt down, I think about the kind of things I want to tell people, also the passion about the microbiome. It just keeps me going. And now looking backwards, it's a really rewarding process, and being able to put out this work out as well.
Oh, all I have to say is that you are, much like the sun behind you, a shining light in research. Microbiome research really needs to have people who are going to take them to the next level. And I really do sense that you are going to be one of the people who's going to do that. Is there anything else that you want to just share based on your research to anybody who might be interested in learning more about this whole idea of infant microbiome?
I would say if you're a parent, like a mom, dad, definitely pay attention to your baby's microbiome. And since your microbiome will influence your baby's microbiome, like we talked about environmental strain sharing, definitely be careful with your own microbiome. And one of the best ways to practice to keep a good microbiome is to eat high fiber food, high fermented food, which is like, this recent cell study showed. And also from my own work, I really witnessed the importance of breastfeeding, breast milk being very important for your baby's microbiome development, and also immune system development.
Well, I cannot wait to see what the future holds. I will be watching you, and I so wish you the absolute best as you continue on. Like I said, you are going to be a shining light for microbiome research, and I'm sure that the people who are watching the show are not going to forget you anytime soon. So once again, thank you very much, everybody. Soon to be Dr. Claire Liu Yu.
Thank you.
Thank you.
Thank you for inviting me.
Well, Ben, I got to tell you something. I have been so fascinated by how microbiome research has been progressing over the years. And in the last, I'd say 2 to 3 years, we've seen some incredible research that's come out, and how it relates to not just our own humanity in our relationship with microbes, but also how we can look to not necessarily the human, because I mean, clinical trials, especially when it comes to poop, it's not always the easiest, and you're not going to get funded all the time. But we do have cousins. And I think that maybe we should be looking at those cousins in order to be able to give us some ideas. Thankfully, well, actually, no, not thankfully, because I would love to go to Kenya, but we do know someone who actually has done this. And so when I first was looking at how we could wrap up this show, I really sort of came upon this particular paper and I thought, oh my goodness, it would be so awesome to have this researcher on, because she has found out that microbes, much like we saw with Claire's data from, mother to child, may actually not only be inherited in that manner, but may actually be inherited based on the environment that is around you. But unlike what Claire has done, the person we're about to talk to has done it in baboons, which to me is unbelievable and is why I needed her on the show. So I want to welcome to the show the most awesome postdoctoral fellow you're ever going to meet out of the University of Minnesota, Dr. Laura Greenison. Hi.
Hi, Laura.
Hi guys,
Hi,
it's great
Laura.
to meet you.
How are you?
I'm doing well.
Thank you once again for joining us on the show. And I'm serious, when I saw the article, I was just completely and utterly fascinated. And the thing that I found so amazing was that you were doing this type of work, which for all of us, we were thinking we need to do either in hospitals or going into different communities, remote and urban, and doing all of this. You went to Amboseli National Park in Kenya. It's like, that's amazing. So before we go into any of your work, can you sort of give us an idea of what that felt like, what that was like going into this type of a habitat to do this type of research?
Yeah, sure. So I did my PhD studying the wild baboons in Amboseli National Park in Kenya. And this is a really cool project because it's called the Amboseli Baboon Research Project. They've been studying the same baboon population for 50 years now. So Drs. Jean and Stuart Altman started studying these baboons in 1971 with this idea that if you set up a long-term field site, you can get an idea of how an animal's sort of ecology and behavior and social environment change over time. And you can look at different life histories. So you can look at sort of what predicts like how long baboons live or die. You can look at their social relationships and how these change over time. So you can collect this amazing data set. And so what that meant for me as a graduate student joining Dr. Beth Archie's lab at Notre Dame is that I got to fly out to Kenya and go to this national park to do research, which was extremely cool. And Amboseli National Park, it's right near the Kenya and Tanzania border. And so you can look up and you can actually see Mount Kilimanjaro. So to study the baboons, there's a full-time Kenyan field research team who lives there. They do the majority of the animal behavior observations year-round. And so they're scientists from various universities of the US and also with the Kenya Wildlife Service and also at University of Nairobi, who are also doing work here. But this field team is sort of like the core, collection group. And so, we'd get up at before dawn, get into the Jeeps, drive out, find the baboons, because there's a few baboons that are radio collared in every social group. And so remember the field team would be out with their sort of antennas standing on top of the car, looking to detect the signal. And then you could sort of watch the sun come up and you'd see like Mount Kilimanjaro and there's like elephants out and giraffes and you're drinking chai. And it was just this really amazing experience. And then the other thing that's very cool about sort of seeing animals out in the field is so these baboons, they're individually recognized by the field observers. So at any given time, the project is following about 5 baboon social groups, and social groups have 20 to 100 individuals or so. And the members of the field team, they individually recognize every single baboon in the group. So kind of like you can recognize like your coworkers or your friends, they can recognize these baboons sort of by the angles of their tail and the way that they walk, and like they'll have like distinctive fur patterns. I can recognize like maybe 5 of them. And so, but that's okay, because I was just out there to collect fecal samples. So, you know, I could, if I saw a baboon poop, then I would tell the observer, I'd say, who's that one there? I see.
So how
donated a fecal sample to the project.
Oh my goodness. Oh, I could talk with you forever about this, but we do need to focus on the microbiome. So please.
Right.
So you have baboon poop, which to me is still like the most fascinating thing. How do you get microbiome information from that? What's the next part of the protocol?
Yeah, so with the baboon poop, so if you collect fecal samples and you could get them into ethanol within about 15 minutes, that preserves most of the bacterial communities, the microbial community. And so we do that and then ship them back to the US and then do for doing DNA extractions. And something that's cool with this project is the way that you preserve fecal samples. If you want to look at like baboon genetics, so how related the baboon individuals are for each other, turns out it's the same way that you preserve fecal samples if you want to look at microbial genetics, so how similar their gut microbes are to each other. And so that means with this project, they have decades and decades of fecal samples, the freezer that were collected to use baboon genetics, 20,000 samples. And it just so happens that we could also use this for microbiome work. So when microbiome research started to become a thing, you know, a lot of the PIs on the project were like, oh, well, let's get together. If we get a whole bunch of grant funding, we can sequence the microbiomes of 20,000 baboon samples that are from these individually recognized baboons that we have all this life history information from them. We know who they're related to. We know, you know, sort of who they socially interact with, so who their friends are. We know what their diet is. We know what's happening in the environment at the time. And so there's just this incredible longitudinal data set that really doesn't exist in humans yet and doesn't exist in many other species, because microbiome research hasn't been around long enough that we have like 40 years of fecal samples, you know, from an individual person or something.
And that to me is just unbelievable, because that brings up 2 very, very important questions. One is that heritability, like I talked about off the top, but more importantly, that means that you can get a really good perspective as to the genetics and whether or not that plays an influence on how that heritability goes, or whether it really is environmental, like Claire, who we just talked to, actually showed us. I have to ask you, what is it?
Right. So like anything, whenever you're asking a nature versus nurture question, the answer is always, well, both, but the environment's playing a much stronger role. We found, and part of that is, so as Claire was discussing, the microbiome changes a lot over time. And so a lot of human and other studies that have looked at this idea of heritability are based on just a few time points. And because the microbiome is just fluctuating, you know, on the scale of hours and days and weeks, depending on what you're eating and where you are and what you're doing, and if you're stressed out or not, it can be hard to find sort of these overall genetic signatures. But if you have, you know, up to 100 samples from an individual over their lifetime, and you have this over several hundred individuals, you can sort of tease apart, like say, okay, well, controlling for these changes in diet and controlling for social relationships and controlling for who your mom is, because as Claire pointed out, right, we do see maternal transmission. Do we still see that individuals who are more closely related to each other have more similar microbiomes? And we found out that yes, that is the case for most of the microbial taxa that we tested. But this genetic variation is like very, very small compared to environmental variation. So on average, although we did find that almost all the microbes have this element of heritability, so they have this element of host genetic effects, host genetics is only explaining like 6.5% of the total variation of the abundance of each microbe. So this paper, right, in a way, it's very cool because it's consistent with what we know about the microbiome, is that there are these overwhelming environmental effects. But it's neat because we could find sort of this low persistent underlying genetic signature. And the reason that that's interesting is it suggests that when we have selection on sort of the baboon genome and potentially on the human genome, we also are seeing natural selection that could be exerting influence on our microbiomes as well. So, right, so you have the selection on the host, it's also sort of manifesting also as selection on your microbes. which is just a very neat concept,
That
right?
that
That these are
is
sort of part of you, but not part of you.
somehow I would just love to see you and Claire work together. Now, when I did some research into your background, I actually came across something that for me was very important. You know, I am of a you know, of a minority in terms of who I am. I know that I have my own health concerns and that I actually have to worry about my microbiome. And that's one of the reasons why I went into microbiology in the first place. An adenovirus can kill me, like seriously, forget about COVID. And when I saw something that you're involved in, I had to bring it up because I want you to talk a little bit about it. It's called the Microbes and Social Equity Working Group, or MSE. And from what I understand, it's about bringing equality across the board of humanity. Now, you're already talking about this from the baboon perspective. We've done a lot of non-human primate translation into humans, but maybe we can do this in a social perspective as well. So I'm wondering if you can just give us a little bit of background on MSE and tell us, you know, how the audience can gain more information about this.
Sure. So MSC is the, as you said, it stands for the Microbes and Social Equity Working Group. And this was founded by Dr. Sue Ishak at the University of Maine in, I think 2018, but it may have been 2019. And the idea is that, right, so there's a lot of us who are studying the microbiome out there, and we would like our research, you know, to be used and sort of applied to looking at sort of human health concerns and human social equity concerns. But that's not really like our field. Like I'm coming into this from like ecology. I don't really know how what I do could necessarily apply to social policy. Sort of make these meaningful social changes.
Let me just cut you off right there. What you are doing will inevitably have perspective on policy development. I've been involved in policy development. It is amazing where we pull stuff from. But yes, sorry, continue.
Right. Okay. Well, I think the idea, right. So I guess it could have, you know, policy implications at some point, but the idea of this group is to say, okay, well, how can we sort of link policymakers with microbiome scientists, like in a more immediate sense? And so one way, if you want to learn more about this group, is that we do have, there's a mailing list and there's a website, which I don't know if I can send you the link and you could post that with the show or something. And there's a group newsletter. And we also have a journal collection coming out. So a whole issue devoted to this idea of looking at microbes and social equity, and talking about sort of 20 questions in the field. And so one of the things, the major questions in the field is something that, you know, almost Dr. Liu was talking about, is that do we see, how do these early life influences on the microbiome, and especially sort of these early life social influences, manifest over time? And specifically, for example, if you have sort of early life adversity, do we see a signature of that in the microbiome? And so one example of this, that this is actually the reason I got into the MSc group and interested in the MSc group in the first place, is I saw a talk a couple of years ago by Dr. Rashonda Jones out of California, that she was looking at, right, okay, so she was looking at how you're feeding infants. So if you're using formula versus breastfeeding, and she found that where she was, that the formula that was covered by WIC was this formula that had higher amounts of corn syrup solids and higher amounts of sugars in it. And so that meant that if you were getting, right, if you were feeding your kids on formula and you had to use WIC, that you're giving them formula that it could actually be setting them up to have more trouble with diabetes or obesity later in life, because it has this higher sugar content. And so her explaining that to me, I was like, oh, wow, this is like a very concrete example of how, you know, microbiome research and policy research should be combined. And sort of the goal of this group is to find, you know, both other scientists who are doing that work and other policymakers who are interested in that, and sort of, you know, trying to move forward.
I'll tell you one thing. We don't have much time here, but I'm just going to share this and then ask you a question. I remember being at a conference after giving a keynote and having a company, a multinational company, coming up to me going, "You're going to hurt our sales because you told them not to have blank." And I'm like, sorry, not sorry. We do need to have that equity, absolutely. Now, you have a perspective unlike any other researcher I've ever met. And so what I want to ask you is, let's go back to what my first talk was about, which was our relationship with microbes, and that can have a relationship amongst ourselves. How do you see your work with baboons with 50 years worth as being influential in how we can identify with our relationship with microbes and, of course, with each other.
Right. So I sort of see it as fitting in a way into the overall sort of One Health concept. So sort of the health of humans, the health of the environment, the health of the other non-human animals and plants within the environment are all sort of the same thing. And by looking at the microbiome, we can sort of extend this to even the microorganisms within the environment. So if we understand what's happening in wild animals, so if we understand what's shaping their microbiomes, and especially how these microbiomes are changing with changing environments. So this could be both like seasonal changes or diet changes or sort of bigger impacts. So we're seeing the climate changes causing these more severe environmental shifts. And so if we can see how that's affecting microbes and by extension, the health of individual organisms, that'll help give us sort of insight into the future to how that could affect human health, other aspects of environmental health, because, I study animal-associated microbiome communities, but there's a lot of microbes that are associated with plants and the environment and crops and other aspects that as we see climate change happening and more environmental change happening, we're probably going to see some shifts in those microbes too. And that might not be ideal.
Wow. Well, I wish you the best of luck. And if you ever need someone to just come out and help you scoop some poop out in Ambaselli, I'm here.
I feel like I haven't actually been out to the field now since 2014, because once a lot of the samples were collected, my whole postdoc, I've just been in the lab, and I'm like, one day I'll get to, go outside and do outside science again.
We'll go have some kafta in Sultan Ahmed. How's that sound?
That sounds great.
Anyways, ladies and gentlemen, Dr. Laurel Greeneisen, thank you so much.
Thanks.
Thank you. Thank you very much indeed, Laura. And thank you to Jason. It's been an absolutely fantastic session. Thank you for hosting.
Absolutely. And I want to give you a special shout out both to you and also to Joseph Shuttleworth for making this day possible. Microbes are everywhere, but you two simply have covered it all. And anyone who's ever listened to my show knows that I want everybody to just enjoy the rest of the presentations, enjoy microbial diversity. And as always, well, make sure to show them some sass. Good night, everybody.
Thanks, Jason. See you next time.
Bye.